What is agglutination? Agglutination is the clumping or aggregation of particles, for example, the clumping of cells such as red blood cells or bacteria in the presence of an antibody. The antibody or other molecule binds multiple particles and joins them, creating a large complex. There are different types of agglutination reactions, but I will be discussing two types. The first type is called hemagglutination. You must be asking yourself, what is the purpose of this reaction, right? Well, here goes. It is used for blood typing. Blood type can be determined by using antibodies that bind to the A or B blood group in a sample of blood. In this procedure, cells and serum of the unknown blood sample to be tested are separated. Cells as well as serum grouping is done. In cell grouping, 1. You prepare a cell suspension in saline from the unknown blood sample. 2. Take 3 test tubes labeled capital A, capital B, and O. Prepare 1 drop of anti-A in test tube A, anti-B in test tube B, and anti-AB in test tube O. Reason for doing this is that in this reaction the antigen is part of the surface of the red blood cells, which has been coated with antigen. Antibody added to the suspension of such particles combines with the surface antigens and links them together to form clearly visible agglutinates. 3. Prepare 1 drop of red blood cell suspension in each test tube. 4. Leave to incubate for 15 minutes at room temperature. And 5. Check for granules. If there is, then agglutination has taken place. In serum grouping, 1. You add 2 drops of unknown serum in test tubes labeled small a and small b. 2. Prepare 1 drop of A cells in test tube A and B cells in test tube B. 3. Incubate for 15 minutes at room temperature. And 4. Look for agglutination. In conclusion, antibodies that bind the A blood group are added and agglutination occurs. The blood is either type A or type AB. To determine between type A or type AB, antibodies that bind the B group are added and agglutination does not occur. The blood type is AB. The second type of agglutination is latex agglutination. The purpose of this agglutination technique is used to determine antibodies such as those produced in response to the rubella virus or the rheumatoid factor. In these procedures, an antibody or antigen coats the surface of latex particles. When a sample containing the specific antigen is mixed with the murky appearing synthesized latex, it causes visible agglutination. The simplest method of attaching proteins to the particles is by passive absorption. Latex and proteins are mixed in an appropriate buffer solution, allowed to reach an equilibrium, and washed several times until the latex is judged to be free of any residual unabsorbed protein. An example of the latex agglutination test is the latex fixation test for rheumatoid factors. 1. You set up 3 test tube racks for each test serum. 2. Antigen must be prepared and added to each tube. 3. Add the test serum to tube 1. Mix and transfer 1 ml to test tube 2. Mix and transfer 1 ml to test tube 3 and discard 1 ml from this tube. 4. Shake and incubate the tubes at 55 degrees Celsius for 2 hours. 5. Centrifuge at 2300 RPM for 4 minutes. And 6. Read for agglutination. Any agglutination in tubes 2 and 3 is considered positive.